The overall goal of this procedure is to produce high-quality crystals from initial crystallization screening conditions. The crystals can be used to study the structures of proteins, DNA, RNA, and their complexes.
Solving novel macromolecular structures has become increasingly challenging. Employing a broad variety of initial conditions increases the yield of useful crystals while reducing the need for sample and crystal optimization. The two fully automated systems, developed at the LMB, facilitate the preparation of up to 1,920 vapor diffusion experiments in nanoliter droplets. For brevity, it is assumed that users are already familiar with the basic functions and maintenance of the nanoliter dispenser, the inkjet printer, and the adhesive plate sealer.
Prior to performing the procedure, ensure that the system's robots are initialized and their controlling software is open. Turn on the chiller of the tube cooling carrier about 30 minutes before the main program will be started. Place a test plate on the motorized SBS carrier of the plate sealer. Run the plate sealer and check that the film is properly applied. Repeat this test twice more to verify that the plate sealer is ready. Then add 20 liters of deionized water to the main container of the liquid handler. Disconnect the coupling inserts from the smaller container of 20% ethanol rinsing solution and connect the inserts to the main container. Enter the appropriate screen name on the inkjet printer touchscreen. Then open and close the carousel door to trigger the carousel rotation.